So we will be talking about the tools of bioinformatics. Now, bioinformatics tools are software programs that are designed for extracting the meaningful information from the mass of molecular biology or biological databases, and they are used to carry out sequence or structural analysis. The pace by which scientific knowledge is being produced and are shared today is at an alarming rate. Different areas of science are getting closer each day, getting closer to each other to give rise to new disciplines. So, bioinformatics is one of such newly emerging fields which makes use of computer, mathematics, and statistics in molecular biology to archive, retrieve, analyze biological data. So, bioinformatics is getting popular due to the inability to analyze huge amount of biological data quickly and cost effective. So bioinformatics can actually assist a biologist to extract valuable information from biological data, providing various web and computer-based tools. And majority of these tools are freely available. So that is the keyword there, freely available. We're going to look at the number of DNA and protein sequences in public databases. So their number actually, they're very large. And the basic steps to searching a database involves aligning the query sequence to each sequence in the database. So BLAST uses heuristics to align a query sequence with all sequences in the database. And the objective is to find high scoring ungapped segments among related sequences. So the existence of such sequence segments above a given threshold indicates pairwise similarity beyond random chance. So this helps to discriminate related sequences from unrelated sequences in the database. So BLAST is Basic Local Alignment Search Tool. So the acronym BLAST stands for Basic Local Alignment Search Tool. So BLAST is popular as a bioinformatics tool due to its ability to identify regions of local similarity between two sequences quickly. So aside that, BLAST calculates an expectation value which estimates the number of matches between two sequences, and it also uses the local alignment of sequences. And we're going to look at the variants of BLAST. So we have BLAST N, BLAST P, BLAST X, T BLAST N, and T BLAST X. So BLAST N compares nucleotide sequence with nucleotide sequences, and BLAST P compares protein sequences with protein sequences. So N denotes nucleotide, P denotes protein sequences, and BLAST X compares nucleotide sequence against a protein sequence, while T BLAST N compares the protein sequences against the 6' translations of nucleotide sequences, and T BLAST X compares the 6' translations of nucleotide against the 6' translation of protein nucleotide. So these are basically the variants, and this is the interface. When you go to the BLAST site, you can see that the BLAST finds regions of similarity between biological sequences. The program calculates nucleotide or protein sequences to sequence databases and calculates the statistical significance. So, and these are the various types of BLAST that you have: nuclear BLAST, BLAST X, T BLAST N, protein BLAST, and so forth. So you can see a whole array of BLAST functionality on this site. Now, so another site we can look at, another bioinformatics tool that we can look at is the FASTA tool, and you can find that at ebi.ac.uk/tools/SSS/FASTA. So it's a database search, and it has a suite of programs for search nucleotide or protein databases with query sequence. Now, when you go to that site, you're going to be presented with this interface. So you can see here that the protein similarity search is a tool that provides a sequence similarity search against protein database using FASTA suite of programs. So this provides the heuristic search, the protein query, and of course, these are steps that you need to take. Select your database, step 1, and of course, you can follow the prompt as been provided. Now, so this is a comparison of the Blaster and the FASTA tools, and both of them, they use a heuristic word, and they are for fast pairwise sequence alignment. So both of them are two similarity search programs, and that helps you to identify homologous DNA sequences. And then they also provide facilities for comparing DNA and protein sequences with the existing DNA and protein databases. So the main difference between the BLAST and the FASTA is that BLAST is almost involved in finding of ungapped locally optimal sequence alignment, whereas FASTA is involved in finding similarities between less similar sequences. So going further, we can look at other two that we can find at www.cluster.org. So this is Cluster Omega, and this is a sequence alignment tool. So it aligns 3 or more sequences all together, and that can help you to find conserved sequences, understand phylogenetic relations. When you go to this site, of course, you can find the two forms of Cluster Omega. This is Cluster Omega and the Cluster W, Cluster X. Now, another bioinformatics tool that we can use is the GROMACS. So GROMACS is a molecular dynamics tool, and GROMACS stands for Groningen Machine for Chemical Simulations. And of course, designed for simulations of proteins, lipids, and nucleic acids. So if you go to the site www.gromax.org, you can find all about GROMACS in here, and you can also look at the functionality of GROMACS. And that takes us to another good bioinformatics tool that we can use, and that is Assemble. So Assemble is a genome browser. And what's a genome browser? It's a graphical interface for displaying information from a biological database for genomic data. So Assemble browser helps the researcher to visualize and browse entire genomes. So of course, it can actually give you the opportunity of predicting gene and proteins, expression regulations, variations, and comparative analysis. So it's another good tool that you can use for mathematics. And I hope that you're going to look at this site and do a course survey of the tools that have been presented tonight. Now, if you go to that site, you're going to be presented with this interface for the Assemble tool. So all we're doing is just doing a course overview of those tools that you can use for bioinformatics. Aside the Assemble tool, you can also have the Genome View, and you can find Genome View at genomeview.org. So Genome View is also a genome browser, and it's a next generation standalone genome browser. So it provides interactive visualization of sequences. You can annotate, and of course, you can also map synthetic factors, short read alignments, and more. So basically, this tool enables visualization of the whole genome alignment of dozens of genomes, and it's a very nice tool for you to consider when you're doing bioinformatics. All right, so if you go to this site, genomeview.org, okay, you're going to find more documentation about Genome View and how to use it. So you can see here there's a next generation standalone genome browser and editor. Now, aside that, we also have a tool that is good for protein modeling. That is a Swish model. So it's a fully automated protein structure homology modeling. And the essence of this tool is to make protein modeling accessible to all life science researchers worldwide. Now, if you go to that site, swishmodeling.expansy.org, you'll find all the documentations, you'll find an array of tools that you can use on the site. All right, that takes us to another site that can be used for sequence alignment, and that is the T-Coffee. T-Coffee is used for sequence alignment. So it's a multiple sequence alignment program. So what this does is that it allows you to combine results obtained with several alignment methods. So we can use it for evaluating and manipulating multiple sequences of DNA, RNA, and protein, and also the structures. That is T-Coffee. T-Coffee. Now, if you go to the site, www.ebi.ac.uk/tools/MSA/T-Coffee, of course, you will be presented with this interface. The sequence alignment program, and its main characteristics is to allow it you combine results obtained with several alignment methods. So this is step one. Of course, they are laid in steps. So going from step one, step two, you can actually see the functionality of T-Coffee. And we're going to look at another tool that can actually give you visualization, and that is the PyMol. So it's for visualization. So if you go to this site, pymol.org/2, then of course you are going to see the PyMol interface. So it's something that is worth trying out. All right, so I believe that this is just like a short introduction of some of the tools that you can use in bioinformatics. So thank you very much.